Every living organism is composed of chemicals which can be either elements or compounds. The elemental composition of living tissues can be determined by following a technique known as elemental analysis. Interestingly, we obtain similar elements while doing an elemental analysis of different living and non-living things. For example, we find that elements such as carbon, hydrogen, oxygen are common to the Earth's crust as well as living tissue. However, the composition of these elements is much higher in a living organism compared to the Earth's crust, as shown in this table. To ascertain the chemical composition of organic compounds in a living organism, we perform chemical analysis. For example, to find the chemical composition of a living tissue, such as a vegetable, we first grind it in trichloroacetic acid. As a result, we obtain a thick slurry. Now we strain this slurry through a cheesecloth to obtain two fractions. One fraction is the filtrate or the acid-soluble pool, and the other fraction is the retentate or acid-insoluble fraction. The acid-soluble pool contains organic compounds. Once a compound has been extracted, the next stage is to separate one compound from the others using separation techniques such as chromatography and centrifugation. Thereafter, various analytical techniques are used to identify the molecular formula and the structure of the compound. All the carbon compounds we derive from living tissues are called biomolecules. For example, carbohydrates, lipids, amino acids, water, and nucleic acid. Biomolecules such as amino acids and sugars are also known as metabolites because they are intermediates and products of metabolism. They are classified as primary and secondary metabolites. Compounds such as hydrogen, carbon, oxygen, nitrogen, sulfur, and sodium, which are involved in normal growth, development, and reproduction, are called primary metabolites. They are found in animal tissues. On the other hand, compounds such as alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigments, scents, gums, and spices are known as secondary metabolites. They are found in plant, fungal, and microbial cells and perform an important ecological function. Some of them play a helpful role as antibiotics and pigments. Living beings are also composed of inorganic compounds such as sodium chloride and calcium carbonate. To analyze the constituents of inorganic compounds, we carry out the following process. We take a small amount of living tissue, such as a leaf, and weigh it. This weight is known as wet weight. Then we dry this leaf to allow the water to evaporate to get its dry weight. Now, when we burn the leaf, the carbon compounds get oxidized and the leaf releases carbon dioxide and water vapor. The remaining ash contains inorganic elements such as calcium, magnesium, and potassium. Therefore, Analysis of compounds helps us identify organic and inorganic constituents in a living tissue. It also helps identify functional groups such as aldehydes, ketones, and aromatic compounds. In biology, however, functional groups are present as amino acids, fatty acids, and nucleotide bases, together known as biomolecules. Amino acids are the building blocks of all proteins. The amino group and carboxylic group are present on the same alpha carbon atom of the amino acid. Hence, they are also known as alpha amino acids. Amino acids are substituted methanes. There are 4 substituent groups occupying the 4 valency positions. These groups are hydrogen, the carboxyl group, amino group, and a variable group also known as the R group. Based on the nature of the R group, there are many amino acids. However, there are 20 standard amino acids in proteins. For example, glycine, in which the R group is hydrogen, alanine, where the R group is a methyl group, and serine, where the R group is a hydroxymethyl group. The chemical and physical properties of amino acids are essentially of the amino, carboxyl, and the R functional groups. As amino acids contain both the amino group and carboxyl group, they can be acidic, such as glutamic acid, basic, as in the case of lysine, and neutral, like valine. Similarly, there are aromatic amino acids such as tyrosine, phenylalanine, and tryptophan. A unique property of amino acids is the ionizable nature of the NH2 and COOH groups. Amino acids behave as both bases and acids. In solutions at a particular pH, ions with both positive and negative charges are equal. This form is known as the zwitterionic form. Another group of biomolecules is fatty acids. Long chains of fatty acids and their derivatives lead to the formation of lipids. Lipids include a broad range of molecules such as fats, waxes, fat-soluble vitamins, and glycerides. They are generally insoluble in water. Fatty acids that form lipids have a carboxyl group attached to an R group. The R group could be a methyl, ethyl, or a higher number of CH2 group with 1 carbon to 19 carbons. For example, palmitic acid has 16 carbons, including a carboxyl carbon. Fatty acids can be saturated or without a double bond. They can be unsaturated or with one or more C double bond C. Glycerol or trihydroxypropane is a simple lipid. When fatty acids combine with glycerol, they form esters, also known as glycerides. Glycerides can be monoglycerides, diglycerides, and triglycerides. Based on their melting points, glycerides may be either fats or oils. Oils have a lower melting point and hence remain as oil in winter, while fats freeze easily at low temperatures. Lipids that contain phosphorus and a phosphorylated organic compound are known as phospholipids. They are found in the cell membrane. Living organisms also have many carbon compounds with heterocyclic rings, such as adenine, guanine, and thymine. These compounds are called nucleotide bases, and they are important biomolecules. When carbon compounds with heterocyclic rings are attached to a sugar, they are called nucleosides. For example, adenosine and thymidine. On the other hand, if a phosphate group is also found esterified to a sugar, they are called nucleotides such as adenylic acid and thymidyllic acid. Nucleic acids such as DNA and RNA consist of nucleotides. They are molecules involved in heredity. Thus, biomolecules act as building blocks of life and perform vital functions in living organisms.